Welcome everybody to the fifth Neuro Talk Evening. Today with Professor Dr. Niels Birbaumer. And today's topic is brain-computer interfaces, so BCIs, application and danger. But before we start today, there is a minor thing we would like to mention. But therefore, I will give or hand over to my friend Aska, who will give you a little advertisement for one of our other projects.
Yes, thank you. I'll make it short because I know you're all waiting for the talk with Professor Birbaumer. So we'll just quickly want to announce a new upcoming event that we're organizing. It's called the Neuro Students Conference. It's an online conference which will take place on the 25th and 26th of October, and it's supposed to be a platform to give for students to present their research because we believe that good research doesn't have to wait until one starts their doctoral studies. Even earlier research projects can be valuable, but they often get overlooked and do not have a platform to be shared on. So we kind of want to create that platform. So if you're a student want to present your bachelor's thesis, your master's thesis, or any other unrelated research project that you're involved in, that is somehow related to neuroscience, no matter which discipline is your background, this would be a great opportunity to present your work to hopefully very big audience to kind of practice your presentation skills, to showcase the work you've already done, and to connect with like-minded students and gather feedback. For your work. And even if you do not want to present, but just want to listen to the research done by fellow students, or maybe if you're not a student anymore, but kind of are curious what upcoming generations are doing, then yes, this would be a great opportunity for you to just partake and listen to some interesting talks. And you can sign up starting now. Maybe you should do that after this talk, though. We will share more information on that later. Yes. So that's pretty much it about this. If you want to learn more about it, scan the QR code or check our social media channels. And yeah, I'll leave it at that, and then we can start with the talk from Professor Birbaumer.
Thank you very much, Aska. So first of all, who's today's guest? Professor Birbaumer is one of the most known neuroscientists ever around. He got his first PhD or received it in 1969 and afterwards received 4 honorary doctorates. He has been a lecturer and a professor at multiple universities around the world. For example, the elite universities, Pennsylvania State, and also the University of Tübingen. He's one of the field pioneers in brain-computer interfaces and also neurofeedback, which is pretty much related. And his focus is neurological conditions or the treatment of neurological conditions such as epilepsy, stroke, and locked-in patients. And therefore, he has over the years contributed to the health of countless individuals, not only directly, but also indirectly to his research. He's the author to more than 500 scientific papers, won multiple awards, for example, the Gottfried Wilhelm Leibniz Prize, which is the more or less biggest German research prize. And he has created or at least been a mentor for a whole new generation of neuroscientists. And all of that not being enough, he is also the author of several books, and among them the best-selling Your Brain Knows More Than You Think. And to go over to our today's topic, BCIs have many possibilities, or there are many opportunities where we can use them. Enhancement of human mind, currently untreatable diseases, and BCIs are actually one of the most promising options. But as always, there are questions we should think about, and one of them is, what are we capable and what is to consider? And with that, I would like to hand over to you, Professor Birbaumer. But also, just to mention to everybody who's attending today, if you don't feel really familiar with speaking English and you would like to ask questions in either English, Spanish, Portuguese, or Italian, Professor Birbaumer can speak all these languages, and he mentioned that he would like to answer them also in your language and then translate the question as well as the answer to all of us in English so that everybody can understand and everybody can ask his questions. Might be interesting and might be an opportunity for you. And also, if you would like to ask questions, it would be kind of nice to show your face. We can afterwards blur it out in the recording if you would like to. But yeah, so enjoy, and therefore there and over.
Thank you. Sebastian. I'm very happy to be here, and I hope you enjoy some of the information I can give you. Anybody who wants to interrupt or to ask questions, you're welcome to do this at any time. If you want, after the talk, if you want to go more in the deep depths of this whole problem, you can read these books which Sebastian already announced. So this is in English, but you can also have it in German. It gives you some information. And if you want to know why I think that BCI is a highly, how should I say, dangerous and problematic type of application of brain science, you can download this book from Lulu Bookstore. It costs nothing. And there I explain most of these political and ethical problems, which we cannot discuss in the short time we have here. Historically, the whole research, at least on my side, started, as Sebastian already mentioned, with epilepsy. We tried to stop intractable seizures in the 80s of the last century, intractable seizures of patients with epilepsy who cannot be treated with drugs and could not be treated with neurosurgical techniques. So we trained them to perceive their own seizures before these seizures started. And as Sebastian said, we did this by showing them their own electrical brain activity. And as you see on this slide, we train them to produce a brain activity which is incompatible with their seizures. So they learned when they were seizure-free, in the few periods when they were seizure-free, they learned to decrease the activation of the brain areas where the seizures started. Most of these patients were able to learn this, and those who had a 70-80% performance and could control the electrical activity of their brain preceding their seizure, these people were able, also in real life, to reduce the number of seizures and the duration of seizures. And among those patients, there were some, not many, but some, who were 100% successful. So they could increase and decrease their electrical brain activity on command in all cases. So we interviewed this patient, asked him how did he do this, how it was possible to increase brain activity and decrease it at will. And it was not clear what these people really did. The only thing which was clear is that they followed the computer, and whenever the computer said, okay, increase or decrease your brain activity, then they were able to do this. Okay, then from that result, the idea that you can use brain activity to communicate started. Here you see the patients. The red line shows an activity before a seizure, and the green line shows the activity after the training, after these patients had learned to decrease their brain activity. That's the green line, indicates a reduction of brain activation. And as you can see, even 6 months and years after they did this, they were able to control their brain activity. They could not describe how they did it, but they were able to control it, and some of them excellently. So then we said, okay, if this is possible, then why not using this capacity of the human mind to regulate your brain activity and communicate with that regulation capacity? And that's why we jumped from epilepsy patients, as I said, in the 70s, 80s of the last century, we jumped to patients who could not communicate, completely paralyzed patients. What you see on this slide is the brain activity of people and the technologies we have at hand right now to record brain activity. And on the left side of the slide, you see the invasive recording techniques, and on the right side, you see the non-invasive. Non-invasive means that we are not opening up the head, and we are not eliminating a bone, and we are not implanting electrodes in the brain tissue. And on the right side, you see the non-invasive technology. Most of the BCI systems which are used now non-invasively using the electroencephalogram, which is the summated electric activity of the upper parts of the cortex. But you can control all types of brain activity. You can use metabolic activity, like with functional magnetic resonance imaging, with a bold signal, so you can increase blood flow and decrease blood flow at will. You can, the same thing is with the so-called near-infrared spectroscopy. This is NIRS here. This is also blood flow. You can increase or decrease blood flow locally at different brain activities if you get feedback for it. fMRI is, how should I say, would be the most interesting one, because with the resonance magnetic imaging, you can go in the depths of the brain. And we did that also, but it's highly expensive, and for clinical reasons and applications in patients, that is not usable. So with the non-invasive technologies, most BCIs available now, also commercially, using EEG or near-infrared spectroscopy. On the left side, you see the invasive technologies, and I will talk about that too. I will describe you a patient where we implanted the electrodes into the brain. Okay, so these are the types you have, and the principle is always the same. You see or you hear your own brain activity, and then the computer asks you to increase or decrease that activity. And if you do it correctly, then you get a reward in the form of a signal, or if you're a child, you get a reward in form of a food or whatever you want. And by that training procedure, by the reward procedure, you learn to regulate activity in the brain, despite the fact that you cannot perceive this activity, neither the electric activity nor the metabolic activity. You can really feel. If you have headaches, of course, out of vascular thing, then you feel it, because you feel the pain, but you cannot feel the blood flow of your vessels in the brain, and you cannot feel the basis of your thinking. And the basis of your thinking is always electrical. And this electrical activity and these electrical potentials of your nerve cells, there is no sensor in the brain who can tell you at what type of brain activity you're regulating right now. So we don't know, or we don't feel what we do here. But that's not an issue. As you know, you can learn many movements, many activities where you don't have a good perception, but you still learn to train. You still, sports is a good example. Many sports, most of these sports, you don't know what you do, but you still can do it excellently if you get the appropriate reward for it. And the same thing is with brain activity. It's like a sport, basically, to learn that. And it doesn't make any difference whether it's single cell activity, or if it's the EEG, or if it's the blood flow. You learn it always and with the same principles of learning, which we know more or less pretty good. Okay, so the main principle is you hear, you look at your brain activity, and you get a reward. You get reward by visual signals or tactile signals, or as you see here, you get reward by movement of an artificial limb. I will show you this in stroke patients, where they learn to move a particular limb or a prosthetic device by just thinking about the movement. And this is very important for clinical applications. Okay, this is for spinal cord patients. It's the same principle. A spinal cord patient thinks about a movement. The electric activity, which is responsible for that movement, is then recorded and transmitted, in this case, to a peripheral device like a phantom limb or another type of prosthetic device. Okay. This person here I show you is a philosopher. It's not a neuroscientist, a philosopher. And this philosopher, Swiss philosopher, Ludwig Hohl, and behind him you see all his statements, which he then summarized in a very interesting book, which is called Notices. And one of these statements was the human being lives according to his communication capacity. losing the capacity for communication means losing life. And when I read this, I didn't know how true and important and fundamental this statement is, that if you lose the capacity for communication, and apart from that, you are alive, but you cannot communicate anymore. That's not a life worth having. And these are the type of patients, after the epilepsy patients, we went to these patients. So these are patients who suffer from amyotrophic lateral sclerosis, ALS, probably you've heard of it, like Stephen Hawking, the famous physicist, suffered from it. And those patients who decide to go on artificial respiration, they end completely paralyzed. That means that they cannot move anything, not the eyes, not the mouth, not the limb, not the tongue, nothing. But they are cognitively intact. That means they can think, they feel like you and me. And these are the patients where at the time we thought is where BCI would be a useful application, and train them. And we train them to regulate their brain activity and use that capacity for communication. I'll show you this in a second with a few examples. This is not true, not very new. The possibility of BCI and the possibility of reading thoughts you find in the antique literature. And Aristoteles is a particularly interesting example because he mentioned that when you're losing the capacity to move, then you may lose thinking. So his idea was, that's now 2,300 years ago, he thought that thinking, conscious thinking and also non-conscious thinking, is a consequence of movement, of the activity of your muscles. And if you lose the capacity to use your muscles over a longer period of time, or if a newborn child never learns to use his muscle, then thinking will not appear. There will be no thinking in the brain if there is no goal-directed movement. So movement and the effect of movement is probably responsible for the fact that the brain learns in the first months and years of life to think. But I will come back to this in a second. So that's an interesting idea, which is old, and it's the basis of the application of brain-computer interfaces. So this was one of our first patients, appeared in Nature many years ago. But as you can see, he's not completely paralyzed because he has his eyes open and he looks at the computer. Apart from that, he's completely paralyzed. He's artificially respirated. He has amyotrophic lateral sclerosis. And he learned by self-regulation of his brain activity to select letters from a computer alphabet. So basically, the computer presented all the letters of the alphabet, and whenever the correct letter appeared, he produced a brain signal. In this case, it was an evoked response in the electroencephalogram, and with that response is transferred to the computer. And when the computer gets this electrical signal, the letter is selected. And he has possibilities to delete it and so forth. So I'm not going on the details. So here you see a letter he wrote many years ago. And but to write this letter, he needed at least an hour or more. I don't remember exactly, but it's not easy to do it, and it's not fast because you have to prepare your brain in order to select a letter, which needs time. You have to produce a brain activity which is strong enough to activate the computer. And this needs a lot of concentration. And these patients, usually after half an hour, they are tired. You have to make a pause, and then they continue. So it's not easy. It's like if you do a highly difficult task and you have to concentrate on this. And without that, you make too many mistakes. Okay, so what that shows is that as long as you have some residual movement, you can do this easily. Everybody of you, those who listen, can do this. Usually you need a training of about 10 to 20, 30 hours, depends who you are, but everybody can do this. Now, what you see here now, this is the real thing. These are patients who are completely paralyzed. And so these are patients who have no eye movements anymore. They have no other muscle in their body anymore. They can move. You can see some of them are very young, like this lady on the right side. She has a genetic form of amyotrophic lateral sclerosis, so she was completely paralyzed at the age of 18 already. So you see on this slide also the two possibilities you have if you not want to implant in the brain of these people. You have the possibility to use electroencephalography on the right and the nearest blood flow of the brain on the left. And again, these patients are virtually blind, so you have to give them feedback auditorily. So they hear perfectly well, but their vision is bad, so that most of them, if they are older, they cannot see anymore. So they are all artificially respirated because the respiratory muscles are also paralyzed in this type of disease. Okay, so here you see what you can do. Most of these patients got EEG. I show you a patient in a movie. I hope it works. So this is this young patient, and you can hear in German, you can hear some questions the computer asks her, and then there's a little pause, and then she has to produce a particular activity in her vessels, in her arteries of the brain. So she has to increase blood flow of the brain. And if the increase of the blood flow voluntarily is high enough, then the answer will be yes. The computer will understand this as a yes. So with this non-invasive device, the only thing you can do, you can train people to increase or decrease brain activity. And with that increase and decrease, the computer can select a letter or can select a loudspeaker or whatever your external device you put on. I show you this. I hope you can hear it. Second.
Der Name deiner Mutter ist Margit.
Now you have to wait. Blood flow. Tries to increase blood flow. If it's yes, if it's no, she decreases it voluntarily. That needs time.
Deine Antwort wurde als Ja erkannt.
So the name of the mother was correct. That's a test question. So you want to know whether the system works.
Du hast immer gerne dein Zimmer dekoriert.
Again, you have to wait. If it's yes, she increases blood flow. You see her eyes are completely paralyzed.
Müsste ja kommen.
Deine Antwort wurde als Ja erkannt.
Okay, and of course, I don't go to the whole video, but you can have it. This is another patient. Now, what this slide and the results of several patients shows is that with non-invasive devices like the EEG, a brain-computer interface is able to at least select a yes, an affirmative, or a negative, a no answer. And with that, in principle, you can communicate everything. You can select letters, or you can say, at least you can select the most critical answers necessary for sustaining life. You can ask, you want to continue living, you're depressed, all these questions. I will show you this in a second. In these cases, BCI makes only sense, of course, if quality of life of these patients is high enough, if the BCI is increasing the quality of life of these terrible of this terrible disease, and if it decreases suffering. If it's not, then this whole technology is useless. So here I show you an example. This is a famous ALS patient in the United States, and he had a very bad quality of life. But if you look at the statistics of many patients, then it's surprising that even without the BCI, the quality of life is fairly high, sometimes higher than in healthy people. And we wanted to know, why is this the case? You would think that this is, in such a situation, you're completely paralyzed. You are, for the rest of your life, you are lying in bed and you cannot do anything, and you cannot even look. That is a horror image for everybody. And many of these patients commit suicide or want to commit suicide before they go on respiration. Now, what we found is that, particularly in the patients who are, for a long period of their life, are paralyzed, their quality of life remains good or is even positive. So what you see here, we presented them tones and slides, as long as they can look, with positive emotional content and negative ones, terrible slides and very nice slides, enjoying slides. And what the red lines, these are about 60 patients, and what you see is that ALS patients in the positive have more positive emotions as negative emotions, as normal, as healthy control people, which is really surprising. Now, when you look in their brain while they're looking at these slides, then we got a very surprising results, confirming their subjective information. While people were looking at positive slides, a brain area was activated. This is a functional magnetic resonance imaging. Here what you see is this red point. This red activity shows you an increase in blood flow. And the area of the brain where this blood flow increases is an area which usually is processing positive social information. So what probably happens in those people is that the evaluation, the judgment of positive social interaction, of attachment, of approach, of nice verbal commands, that they get more sensitive to the positive aspects of our social interaction, despite the fact that their social interaction is very limited. All of these patients live at home with their family. So those patients who have no family and cannot live at home and stay in hospitals or homes, they don't want to live. They all died. Okay. Now here look at depression. ALS patients are not as happy as healthy individuals, and at average, as you can see, but they're within the normal range. And psychiatric patients who are deeply depressed and melancholic, they are in a much worse situation than these patients. Again, you see this is another group of patients. You see quality of life. In healthy people, that's blue. Then in ALS patients, that is red. And then in the caregivers, and you see those people who take care of the ALS patients, they are not as happy, of course, as healthy people, but they are not different, again, not different from these completely paralyzed patients. So this is a good news, and shows that BCI is a critical and very important technology helping these people to stay with a good quality of life. Okay, so this is shows also, because as I said, before respiration, many people want to die. If you ask these people before and after a year with BCI at home, then you can see that in patients, the wish to die is much lower than in healthy people first. And healthy people think what you would think too, I guess, and I also did that before I worked with these patients. What everybody would think, oh God, with this disease, it wouldn't be worse to live. It would be terrible. I wanted to be dead. So you see, this is wrong. Don't believe your subjective impression before you measure it, and you see that patients more or less lose the wish to die, despite the fact that their paralysis is progressing. Again, surprising. Well, what is the reason for this? There are many reasons. One reason is probably that what I show here is a meditating monk. The EEG of these patients looks like this of this meditating monk. If you don't have no muscle activity, your muscles are completely relaxed. Also, the brain activity relaxes, and you see these activities waves, alpha waves and theta waves you see here, show that the brain of these people is relatively relaxed. And here the red line in the figure below see that these patients who are completely paralyzed have a much lower brain activity as normal people. So again, this indicates that they're in a, let's say, relatively relaxed but awake state. Also, that may be a reason why in such a state your quality of life is good. Also, when you look at sleep, this is sleep EEG, and what you see when you have deep sleep, then you get, this is what is here. The letter E shows your deep sleep, which you usually have when you're young. You usually sleep deeply about half an hour after you went to sleep. And sleep is the best indicator of depression, because if you're depressed, particularly deep sleep is going away, your EEG is irregular, you wake up all the time, your sleep is very bad, and so forth and so forth. So we looked at the sleep rhythm of these people during the night, and what we found is what you find in healthy people also. You see that look at the blue line, you see that these patients go on deep sleep like anybody else. Then they have a first dream cycle where they dream for about half an hour, and then they again have a deep sleep like anybody else. So their sleep profile and their circadian rhythm is fairly normal, and that confirms their personal and subjective experience that they don't have a bad quality of life. Okay, now what's the problem with this? Well, there's not a real problem, but the problem is, one problem we encountered is that with these non-invasive devices, with EEG or near-infrared spectroscopy, we were not able, or the patients, I should say, the patients, not we, the patients were not able to communicate in a normal manner, in the sense that they can select fluently letters, write sentences, describe their inner state, and all these type of fluent verbal communication. We were never able to train people to do this. Whether this is a question of the disease or whether this is a question of the technology, I don't know. I have no real answer to this. I didn't have the time to investigate the reason why people were not able to select fluently letters and write sentences like we would, what we would like and what the patients also would do. So we decided to go into the brain. And the question was, is it a technological question or a psychological question? And so the EEG measures only the activity of millions of cells, and all the cells do different things. So in order to control this brain activity of millions of cells, it may be difficult because some of the cells do different things than the others, so they are incompatible with each other. So that's why we decided to use microelectrodes where we can record from single neuronal cells. And we implanted a set of 162 microelectrodes in the brain of a patient who was already completely paralyzed. I'll show you this. So this is this type of electrodes. They are not as fine and not as nice as the one with Elon Musk and his company Neuralink produces. Musk's company, at that time when we did that, wasn't really ready. Produces a BCI system which has several thousands of electrodes, microelectrodes in the brain, while at that time we were only able to implant this type of electrodes you can see here. It's very small. It's not dangerous in the sense if you don't get an infection, it's not dangerous to implant it. It's not destroying many cells. And it was tested on many patients before we did that. Okay, here you see these patients and you see the system. So there's a system which analyzes the cell activity of all those 160 cells, and the patient who has these 160 electrodes implanted, and the software, when the software is programmed in such a way that it analyzes the brain activity, and the patient is presented auditorily with a group of letters in a different sequence, and whenever he wants to select the letter, he does something with his cells. Don't ask me what. Patients cannot describe what they're really doing. Okay. What you see here is, again, these letters. They're presented in colors, but for the patient doesn't matter much. In the early days of his disease, he learned that some letters are associated with different colors, so we kept those colors. So we have a group of letters A, A, D, C, B, F, another group of letters with N, S, L in the frequency of these letters. So the patient knows this. He doesn't have to look because he cannot look. And whenever a letter appears which he wants to connect with the word, then he increases the firing rate of these cells. And here I show you what we do every week with these people. We ask them about their quality of life, and the patient has to answer. These are internationally standardized questions about quality of life, and the patient has to say yes or no. And what you can hear, I hope you can hear it, if he increases firing rate, the tone, which also the patient hears, the tone is much more intensive, gets higher in frequency. If he wants to say no, he regulates his cell activity downwards. So then you hear the tone gets lower. It's a lower frequency. And of course, the computer also tells you if it's a yes or a no. And you see the electrodes, the cable of the electrodes comes out of his brain here. Now I show you the question. Some of them, not all of them, just some that you see. It says, du bist selten müde.
Nein.
Du bist oft wach.
Ja.
Du bist selten wach.
Nein.
Du denkst viel nach.
Danke.
Du denkst viel nach.
Ja.
Du denkst wenig nach.
Nein.
Du bist oft traurig.
Nein.
Du bist selten traurig.
Ja.
Du bist selten niedergeschlagen.
Ja.
Du bist oft niedergeschlagen.
Nein.
Du lachst oft innerlich.
Nein.
Du lachst selten innerlich.
Nein.
Nochmal.
Du lachst oft innerlich.
Ja.
Du schätzt dein Leben sehr.
Ja.
Dein Leben ist sinnvoll.
Ja.
Okay, so I'll show you another type of activity while he is.
I'm sorry, Professor
Yes.
Birbaumer.
Yes.
May I ask a small question before we
Sure.
skip slides? Because there was not a real opportunity to ask, but you showed us a slide that indicates or that shows that people with ALS that are totally locked in have actually more positive or negative or emotions at all in reaction to different stimuli. Might that be also or could we interpret it as a coping mechanism because they might be so impaired in their social activity because they can't really choose if they interact with somebody else because people have to come and meet them basically or show around, that they try or our body tries to cope it with getting as much stimulation in terms of emotional stimulation out of it in a single stimulation as possible?
Sure, that's possible. There are many reasons. There are many reasons why the quality of life of these people is so good. And of course, the coping process is in any case at work all the time. If you're diseased, if you have another disease, it doesn't have to be as severe. Automatically, the brain produces a set of coping processes, and you need more social interaction. You ask him for more social interaction, and so forth. So this plays a role, but this is just one part of it. But the main part, actually, and that we looked empirically, is a very simple one. And that makes sense. And a very simple one is quality of life is only acceptable if family or close, let's say friends or persons who are close to you are taking care of you. So all those patients or those patients who have no family or whose family is not taking care of them, those patients learn nothing. So that means their BCI, even if you give them a BCI, their BCI does not work at all. They never learn anything, and usually they die much earlier than others. So this is the main factor, but the factor you're naming, of course, plays a role. There's no question. Yeah, that you're right. Okay, I'll show you now a sequence where he selects letters. Now, what you see, I have to describe this shortly before I start the video because you need to know. So he sees nothing, but you see it. So he hears, he knows when the computer says green, then he knows which letters are in the green. That he knows this from the past of his life, where he learned with us while he was still able to look. He learned to select letters by these colors. So he knows when green comes, it's these letters. When there's red comes, it's another group of letters. And then when he selected the right color, then he starts waiting for the right letter, and then he selects a letter. So it's a long process, but with this he can combine all types of letters he wants. Then there's of course an end signal. He can stop this at any time, or he can start it again, and so forth. So I just tell you this, that what you also see at the beginning very shortly is the surprising fact that in this patient, basically it's only one or two cells which he's using to spell and using to speak, if you want to say that way. So it's not that he's using all these 160 cells of his brain. He has learned to select letters with a particular cell, one or two, not more than that. So obviously, the implantation of so many cells, at least after a process of learning, is not absolutely necessary. Of course, to find that cell who is then communicating, you need many electrodes. Whether you need as many electrodes as this mask system from Neuralink supplies, these thousands of electrodes, I doubt that. And I think most people will learn shortly, even if there are only a few cells, a few electrodes are implanted, they will rapidly learn to select letters and communicate with a few cells as you learn to automatize your thinking process or your sports or whatever you do. Okay, now I show you this. I hope I'm here. Okay. Okay, here this is his letters and the colors of the letters. And on the right low, you see the cells. This is cell 21.
Nein. Sie.
Now he selects on his own will.
Nein.
The computer presents and he thinks.
B. Ja. Der aktuelle Satz ist B.
This is abbreviated.
B.
Okay, the video is not the
Gelb.
original.
Nein. Grün. Ja. N. Nein. Ja,
he was doing this after the operation.
der aktuelle Satz ist Birbau. Ja. Der aktuelle Satz ist Bierbäumchen da. Gelb.
He talked to me
K.
as Birbäumchen.
Ja. Der aktuelle Satz ist Bierbäumchen da blank.
So basically,
Gelb. Ja.
he says thank you for the implantation.
E.
This was shortly after
Ja.
the implantation. And
Der aktuelle Satz ist Bierbäumchen da blanke.
it's not
Gelb.
100% correct, but you understand it.
Nein. Grün. Ja. N. Ja. Der aktuelle Satz ist Bierbäumchen da blanken.
Okay, so and so forth. So the sentence he was spelling here was just he was saying basically thank you for and your team for allowing me to do this spelling or for the implantation. That was the whole thing which he repeated several times. So it was not particular, but he selected all these letters out of his own will. We didn't ask him to say this. He said it out of his own. And in the paper, you can read the paper in Nature Communication, and there's the whole sequences of sentences over several months is published. So you can see what he says and what his wishes are, what his quality of life is. I have to say that the time he needs to spell these sentences is pretty slow. So let's say for a sentence like this was an abbreviated version of the video on of the sentence, he needs about an hour in order to present, let's say, 3 or 4 sentences in a grammatically order which you can understand. So it's not as easy as you would think, but it's possible. Is that clear?
Yes, thank you.
Okay. So I will finish now, if we have some time left, with a few minutes, with another application, which is, at least from a clinical point of view, more significant than ALS, because ALS is relatively seldom a rare disease, but it's not really rare, but it's relatively rare. But what's much more frequent is stroke. And in stroke, as you know, there are some stroke patients who are not completely paralyzed, but virtually completely paralyzed, who cannot move the arm anymore. And even after 1/3 of the patient, even after a long rehabilitation training, their arm and their hand is still paralyzed. Now, before I show you the BCI, which tries to eliminate this paralysis, I'll show you the reason why very often the paralysis is not a consequence of the destruction of the brain, like here in these monkeys. These monkeys were investigated in the US many, many years ago by Taub. And what he showed is that these monkeys, if he deafferentiated, that means if he eliminated the 2/3 of the patients, so what today is the most effective and cheapest treatment for paralyzed hands in stroke is to paralyze with a prosthetic device or with a kind of a device which doesn't allow any movement anymore. So it's called constrained movement therapy. So you constrain the movement of the arm by blocking arm movements of the healthy arm, not of the sick arm. And you force the patient to use his paralyzed arm. And if these fibers are intact, then slowly the patient learns to move again, despite the fact that he was completely paralyzed before. So this is one treatment. However, if a patient has complete paralysis and you stop the movement of the healthy arm, the patient will die of hunger. He cannot move anything anymore. So there's 1/3 of these patients who have no connection anymore after the stroke with their paralyzed arm. So for these patients, it's 1/3 of all stroke patients, and in Germany we have about close to 1 million of patients a year. For these 1/3 of the patient, BCI is the indicated treatment, and I show you, and it's not invasive. You don't need an invasive system. You just need the EEG. So this here shows you how this is working. So the patient has usually, if he cannot move the arm, he has a type of a prosthetic device, and he has to think about the movement. So we ask the patient, move, do this, open your hand, close your hand. Even if he cannot, we ask him to think about this and to try to open and close it. And when you do this with your brain, then what you see here is that a particular type of rhythm is changing. And if this is taking place, then the BCI is moving the paralyzed arm. So the patient thinks a movement, and at the beginning of the training, he has a prosthetic device. And then when he thinks correctly that movement, then the EEG, the brain activity, is moving the arm. And later you slowly eliminate the BCI and allow the patient to do this without the electric assistance here. I'll show you this in a video. Here you see some patients. I will not saying anything because otherwise I'm disturbing your vision, but I guess you can understand it. So these were two groups, one placebo group and one non-placebo group. But that's not important, that's methodology, and then you can see it here. If it's green, if the EEG becomes green, the patient thinks correct about the movement.
Linke Hand. Los. Ende. Linke Hand. Los.
Good.
Ende. Linke Hand. Los. Ende. Linke Hand. Los.
Okay.
Ende.
Okay, now for the hand. Same thing for the hand.
Rechte Hand. Los. Ende. Rechte Hand. Los. Ende. Rechte Hand. Los. Ende.
Okay, then they get physiotherapy, that you need to give them physiotherapy in addition.
Und strecken. Ja, sehr gut. Strecken. 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 Ich helfe mal mit den Daumen abzuschrecken. Jetzt ist es ganz super. Ja, strecken. Sehr gut. Jawohl. Und wieder entspannen. Ganz entspannen. Gut, danke. Hoch. Ja. Und wieder hoch. Und wir streben an. So. Also ich helfe mal mit der Bewegung. So. Und dann hoch. Ja. Und wieder runter.
Okay.
Hoch. Runter.
Okay, now I try to get out of this. move along. And what you can see that is a little bit difficult to understand, but I can tell it. I give you a verbal description. This is the brain activity in the functional magnetic resonance imaging before the training and after the training. And basically what you see is that in the course of the training with the brain-computer interface, patients move their brain activity from all over the brain back to the area of the brain where the movement originates. So they learn, or the brain relearns, to use the right brain areas so at least those who are not completely destroyed, those who are remaining, and there's always some healthy aspects which remain after the stroke, and they learn to reactivate those originally silent brain activities. So what the brain activity, what the BCI does, it trains the brain to activate those cells of the brain who are still intact, but who were not used in the past. And there are many of our cells are silent during the whole life. And by a particular type of training, we learn to activate these silent cells. And that is what the BCI training does. Okay, just to finish that up now, let me say that all these gives us a huge hope, and it shows the positive aspect of the BCI training. Now, let me say some aspects or some problematic aspects of the BCI. Let me get away with this. Oh, sorry, now I am. Can you still understand me? Yes? Now, as you can see yourself, you can do more or less everything with this type of technology. And what I showed you here, you can train brain activity to regulate the external world with your own brain without any movement. But the same thing can happen from the outside. So, for example, we can stimulate the brain electrically, or we can use the BCI to stimulate those areas electrically or chemically. And the BCI probably is also used, and by that we can manipulate, of course, without the will of the patient. Our patients, of course, gave their agreement, they wanted to have a treatment. But when you use this technology externally without the permission of the patient, you can do everything. You can manipulate more or less all kinds of behaviors, and with there's several studies showing that you can increase the performance of BCI by adding electrical impulses to this learning, to these training activities. So when I train patients to do BCI and at the same time I stimulate the brain with an external current, I get a much better result. So I can push people to use BCI training in a much more efficient way, but I can also push brain areas without knowledge of the patient. I could push brain areas to increase their activity and to use a BCI without the knowledge of the patient, because you don't feel your own brain activity. So the possibility of manipulating against the will or against the permission of the patient is endless. And I'm afraid that with the fact that now Europe is very weak in this technology, and China and other countries who are not as democratic as us, or as the European countries, they're moving fast forward, and the BCI is one of the main research topics in these countries. And as you also know, people like Elon Musk, who invested billions in the development of BCI, they are not interested in therapeutic applications. They are only interested in selling huge amounts of these BCIs to healthy people. And that's why I'm strongly advocating to give this technology only by prescription, so that in order to use it, you have to have an illness, or you have to have a disease which cannot be treated otherwise, better. And then, if this is clearly stated by independent doctors, it should be possible to prescribe it. To prescribe it with the same limitations as you prescribe an opiate or a dangerous drug. So you should not allow a broad commercialization of this type of technology for healthy people. Of course, I understand that many healthy people would like to have this, and I understand why many people think they can increase their capacity, which is probably true. Their cognitive capacity can be increased and can be improved with BCI training. There's no doubt about that. But I don't see this as a useful social, political, or otherwise useful activity, and that's why I strongly warn the public and warn everybody who is related to that activity to use it. And we have no idea what you can do with that, but there's no question that it can do enormous danger. That this is highly dangerous and difficult to control technology, which shouldn't be widely distributed. But I'm a minority here. I don't know anybody who has this opinion. Most of the people who work with this think they can make money with it, and I guess that's true. Okay, thank you very much for listening.
So thank you very much, Professor Birbaumer. Were very nice and very impressive insights. And if there are any questions right now, I would like to let them go first. But otherwise, as I've sent to you, there are a few questions from the audience that get in before, and we could go through a few of them if you would like to get more into detail of methodology or the technical details and actual methods. But there is one question, Shaheen.
Hello, Mr. Professor. Hello, everyone. You hear me, right?
I hear you very badly, but Sebastian can translate it to me, I guess.
Okay. So hello again, Mr. Professor. Hello, everyone. Thank you for the presentation. I have two questions, actually. One of them is the resolution. How small should be or how big should be our resolution in order we get more accurate like feedbacks from the brains of the patients? That was my first question.
Yes. Can I answer that right now? I don't think that I can answer the question once and for all, but from empirically, well, this is a very important question. And empirically, dependent of the aim of your research or the aim of the application you have, I don't think there's any doubt that the resolution on a single cell level is the best. So which makes sense, the closer you are to the origin of your brain activity, the better you can manipulate it. So invasive technology is always superior to non-invasive technology first, and the resolution, as you said, let's say in the EEG, the finer the resolution would be, the better it would be. But you know that the resolution in the non-invasive systems is very bad. So EEG, near-infrared spectroscopy, these are the only things which are economically viable. Their resolution is very bad. So even if you have hundreds of EEG electrodes, you cannot even record the activity of a single finger, for example. You cannot discriminate between the activity between finger 1 and finger 2. So how you can train a patient or how you can do this in training a particular body part. But as I showed to you, there are diseases like stroke where it's not necessary to have a great resolution. Who cares what the resolution is in these patients, as long as the patient is able to move the arm with the system, and if he can do this, as you saw, that works and it has a therapeutic value. So the resolution depends on what you want, And then you can decide on the resolution, and then you can decide on invasive and non-invasive.
May I ask my second question, please? Yeah, my second question is about the first part of the presentation about the yes and no answers of the patients. It was like that even when we have sort of algorithm or blood flow, we count it as yes for the patient or no. I just wanted to know if we can prove it, if it was really yes, like in healthy people, it should be maybe different for interpreting a little bit of EEG or blood flow of the patients or of the people.
Well, it's a good question, but the only thing I can tell you, when you look in the literature of the success rates of BCIs in spelling, for example, or moving the arm or moving a prosthetic device, what is surprising to me, when you look at the percentage of people who learn it and how long they need, there is not much difference between the patients I showed you and the healthy people. In healthy people, you have people who can learn this within half an hour. You have other people who need days and weeks in order to learn it. Then you have some people which we call BCI illiterates who will never learn it, and we have no idea why they don't learn it, but they learn other things perfectly well. So there seems to be something special in learning your own brain activity. And the reason may be because your own brain activity is so difficult to perceive. And even if you're trained, I trained it for, I don't know how many years, 20 or 30 years, and I'm very good. In some part of my brain, I can regulate very good. But if you ask me how I do this, I still have no idea, despite the fact that I do it since many, many years. And I'm not using it because I never found out that it has any use. I cannot concentrate better because of that, but some people can. So again, it depends very much of what you want, and it depends what, but I agree with you. But the difference between normal people, healthy people, and patients is if there's not a severely brain damaged person, that's a different issue. We had some patients who were in a coma or pseudocoma or comatic patient, and it never worked with them. It never worked with them. They had huge lesions, and with the brain, large parts of the brain are destroyed. It's not working anymore.
Thank you so much.
Thank you. And there's another question from Sarah and Omar.
Hi, good evening, everyone. Thank you, Professor Birbaumer, for the presentation.
Okay.
I'd like to ask you a question on your position on the future development of the tech and the wide deployment. If you could clarify or elaborate on your position, why this tech, in your opinion, is as dangerous as opioids, and why do you think the widespread of this tech or the commercialization of this tech is a dangerous thing? You did recognize that it could be an enhancement to cognitive functions, to humans in general. So, yeah.
Well, this is a difficult question. Your question is as difficult as my answer. And of course, there's no real empirical data for my opinion. My opinion is based on my imagination, what I can imagine, what you can do with it. And I give you an example. And one example, which I wrote last year in an article, you can read it if you want, is lie detection. And with these type of systems, and with the application of external currents in addition, of course, you can make lie detection 100% correct, in the sense that assuming somebody, you have a suspicion, and you have a person who you suspect to has done some bad things or crime, by implanting an electrode or by measuring the fMRI to particular questions, you can come to 100% correct results. Now the question is that desirable? Do we want that? Do we want to know the truth all the time? Of course, with some crimes, severe crimes, I would agree that it may be useful, but for the rest, it's not useful. I don't see any reason why should we increase our cognitive capacity? Let's say more than we already have it. If you look what we did with our fabulous cognitive capacity over the last centuries, I don't think that we have any indication that if we would have a better cognitive capacity, we would do in any way better. Of course, if I have a patient who has a severe, let's say, problem of cognitive capacities, then we have an indication for BCI. I agree with you. But a wide commercialization, what would that bring to us? What would be the profit for the non-pathological case? And that I cannot see. That's why I think it should not get out of the hands of let's say ethically responsible people. And of course you can, as I said, you can with external currents you can manipulate any behavior you want. And that will be used by dictators, by people who don't want to have responsible decisions from their citizens. That's why I'm afraid of this technology, of my own technology, let's say it that way.
Thank you, Professor. I also do think that it is necessary to set guardrails if we were to make this move. It has a lot of potential. It could be used in multiple fields in a civil way. However, there are risks, serious risks to humanity.
Yes.
And if we don't set the guardrails at an early stage of development, then we could potentially lose control
Yes.
and have undesired consequences.
Yes,
The
that's
widespread or deployments of this tech.
Yes, exactly. That's what I'm saying.
Thank you.
Then there's the next question from Aska.
Yes, thank you very much for the talk. My question might be a bit more on the technical side. So I was wondering about the generalizability of models required for the differentiations of answers of people. So I imagine that for each patient or each healthy participant, you would have to train a separate machine learning model, be it a support vector machine, a neural network, whatever, matching the activity of that specific person. Is there any degree of generalizability in this model? So could like a simple yes/no model to some degree be generalized to a patient from the same conditions, or are the individual differences just too large for that to be possible?
That's a very good question, and the answer is very clear. I didn't say this, and I'm sorry for this. You are absolutely right. Before what you saw in the video is, of course, the end of a long process. And these are deep learning models, and that's the basis of the analysis, as you already said. And for each patient, we have to do about an hour of training period each day again. What's even worse, it's even worse than you said. In order to train the model into a fairly, let's say, I'd say fairly correct detection rate or participation rate, you need, for each patient, even on the next day, it's already different. So every day we have to train new, and with every patient we have to train it again. It may be possible in the future, that's up to you if you want to do that research, but it may be possible to find a generalized model, but we were unable. So we had to spend an enormous long time in order to train the artificial intelligence model to the right thing, particularly with the EEG. With the fMRI, it's easier because it's a slow signal. It's very clear to recognize, but the EEG is a pain in the neck because it has high frequency range. It's different from day to day. It changes with your sleepiness and blah, 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 blah. Now, with single cells, it's much easier because the single cell responds or it doesn't respond. So if a patient is good and he learns rapidly, the learning period of the model is much shorter with single cells than with EEG. But work on it, and then maybe you find a generalized model. Could be with we lose ancient type of algorithm, mostly support vector machines, which are now completely outdated. They are new methodology of deep learning, which we haven't even in mind at that time. So you may be much better today than we were. But remember this, we started this 50 years ago. And artificial intelligence didn't even exist at that time. And at the end, we had support vector machines, and the support vector machine study said yes and no, and that's it.
Thank you very much.
So, are there still questions from the audience right now? Otherwise, I would just go to maybe up to 3 questions that were collected before, so that there are just a few of them are answered. I guess no. So I would just read the questions out to you, and you could answer them if you would like to, Professor Birbaumer.
Sure.
Okay, perfect. I guess the first one is already answered. It is about how promising do you think is the consideration of ethical and legal issues in neurodata? You made a quite clear statement on that. So the second question would be, do you think that brain-to-brain communication, like shown in some experiments by Miguel Nicoleles, could also lead to a better understanding and treatment of locked-in patients? You also mentioned or show a few of his studies and findings. So I guess you're pretty much into the topic.
Yeah, that's a good question. I think it discussed with Miguel Nicoleles personally. He's a good friend. And when he started this, I was impressed, of course, by his studies and that these 2 monkeys learned to cooperatively work on the BCI. So whether this is useful for patients, I don't know, but it relates to the first question and Asuka's question. Since these patients are so different and you have to train the model so long for each patient again, to connect 2 patients, I think will pose enormous methodological problems. With the monkeys, it's easier because when Miguel trained the monkeys to move the hand or the arm, and there we know the brain activity, we know what we have to do, everything is clear. But here we have patients who want to express, let's say, quality of life, or want to say, I want this, I want that, and please come in, please go out, let my mom come in, and all these all of those have different brain activity. And so putting these people together, so far, I don't see any particular sense. It's just not working in methodologically. But again, coming back to the ethical thing, it can be very dangerous because you can now, by putting these 2 BCIs together, you can force, of course, one patient to use the brain activity of the other patient. When I say to the patient, now, increase this brain activity here. Why you think this? Do that. And then I reward the other patient for doing what the other patient did, then within a very short period of time, they will think the same. Do we want that? Of course, I can force people to think the same with this system. By rewarding them, whether they know it about the reward or not, it doesn't make any difference. I can reward them by stimulating a particular brain area. I can reward them unconsciously with a particular change in the slide or whatever I have. And within a few hours, they will think the same. So again, I have my problem with that. I don't want that people think the same. They already do that without any BCI, and it's bad enough.
Thank you very much. I guess the question is answered. If not so, please speak up if the person is still here. But I would just then continue with the next question, and that is there any research regarding not only restoring function of BCIs, but also enhancing them, like extra senses or help to better concentrate, or maybe overcoming any other natural limitation of a healthy brain that I can think of right now, or that we can think of right now?
Yes. Well, I didn't do myself research, but I just can tell you over the literature. There is extensive research now going on the application of some of these currents we are using, particularly slow cortical potentials. So they put the potentials which prepare movement or thinking, they put them from the outside of the head. You put a positive or a negative current on the head, which you don't feel if it's not too strong, and then you feel it. But if it's not too strong, and there's clear evidence that this is helpful also for concentration, and it's also in patients. It showed some positive effects, particularly in stroke patients, to reactivate those cells who are paralyzed by the stroke. So that's just one aspect of it. And I tell you what my impression is, but that's not scientifically proven, just to answer your question. My impression, I couldn't prove that, but my impression was that those patients with ALS who trained for a long period of time with our machine, and we followed them over years, and we have to do this all in their home. We had to travel to these patients and so forth and so forth. My impression was that it kept their cognitive capacity on a higher level, because if you're completely paralyzed and you don't have a capacity to communicate, then what's happening is what Aristotle has already said: you lose thinking. You just stop it, because any thinking doesn't make any sense anymore. Whatever you think, it will not happen. Whatever you want, it will not happen. So PCIs definitely have the capacity to improve, or at least to stabilize, your cognitive capacity. And when you combine it with external currents, and from PCI you know which current in which patient would work at which brain area, then you will improve performance. But as I said before, I don't see any sense in the long run in doing this with healthy people. But you may have another opinion.
Thank you again. And then I guess the last question of the evening, then would it be perfect landing. There it is. How could 100 Hz clock cycle EEG-TMS, so OI loop, achieve a non-invasive BCI with a human brain in the loop?
Oh yeah, that's I think also Nicolelis tried this, but that's a very good question. And if you put external current in the loop, that is increasing the performance of the PCIs. We didn't do that for many, many different reasons, complications, ethical reasons. But I agree with you, when you put, like, you have a particular, you want to increase the activity of a particular cell or particular area of the brain, and then you transfer your current, which you get out of the brain, you transfer it first to an electric stimulator, and then a stimulator stimulates by the lead of the artificial intelligence algorithm, stimulates the brain area which is responsible for the increase of that activity or that performance. If you do that, then you will improve, the system will improve rapidly. And, you know, sometimes we had spent years with one patient before we got any useful answer. And so this process can be abbreviated probably dramatically with that idea. So I agree with you. And in the future, I think this is a future way to go, but it has to be tested scientifically, and that is not done right now.
Thank you very much. And so I would say this could be the last question, if not anyone else would like to ask questions at the moment. I guess I wait like a few seconds. Otherwise, I would just end the evening with the rest of our presentation, but this is just the casual advertisement of the next events. As I see, or as far as I see, there are no other questions. So thank you very much, everybody that attended, and also Professor Birbaumer. It was a very, very interesting call.
Yeah, have fun.
Thank you very much.
Okay.
And the next Neuro Talk evening will be on the 9th of October with Professor Gruber about gamma band oscillations in the human EEG. If you would like to read more about Professor Birbaumer, you then can look at his links up that are listed right here. And also, if you would like to know more about our network, you can, as always, just scan the QR code. And yeah, maybe you will show up at the Neuroscience conference. We would really enjoy, and it would really be a pleasure if a lot of people would show up there, and we have a great day or 2 days together. And yeah, with that, thank you very much. Have a nice evening, and see you next time.